Working? Yeah, talk today. Yesterday. Yeah.
Yeah.
You good to go?
All right. Thanks for coming today. Before introducing our speaker, let me remind you of the events of the coming days. On Fridays, there is no lunchtime talk, but we are sponsoring the talk organized by the Department of History and Philosophy of Science, given by Jim Peebles, Nobel Prize laureate in physics from Princeton, who will be talking at 3:30 p.m. on the 10th floor on the physicist's philosophy of physics. So if you want to hear what Peebles, Nobel Prize laureate in physics, has to say about how physicists think about physics, please join us at 3:30 p.m. on
Just to say, despite the title, I think this is very much a philosophy of science talk. It's about scientific methodology, not the kind of content-specific theory stuff that we often call philosophy of physics. So I think the interest could be quite. I encourage people, even if you're not a philosopher of physics, to consider attending.
Very good. Thanks. Thanks for the clarification. And it may be, because it's hard to predict, that the room may fill quite quickly. The physics department is aware of a Nobel Prize laureate coming to Pittsburgh, so that might draw a few individuals. Hard to predict, really, but you might want to come 5 minutes early just to be on the safe side if you plan to attend Peebles' lecture. On Tuesday next week, we have another lunchtime talk given by Mike Dietrich, who will be talking about species diversity, and the title is "Does Species Diversity Matter for Biological Research?" And on Friday, one of our two postdoctoral fellows, Alia Dewey, who is right here, will be talking about reformulating rational analysis, so a talk in the philosophy of cognitive science. That's not
It's
the title.
changed. Design explanation and behavioral neuroscience.
Design explanation and behavioral neuroscience. Same field, however. That I got right. All right, today it's my pleasure to introduce a friend of the university, the center and HPS, Jim Tabery, who is a professor of philosophy in the Department of Utah. He has also an affiliation in the Department of Internal Medicine at the University of Utah. Jim is a historian and philosopher of biology. He has written extensively about the history and philosophy of genetics, including its recent implementation in modern biomedicine. He has written extensively and published in many journals in philosophy, such as Philosophy of Science. He's written the recent article in the Stanford Encyclopedia of Philosophy of Genetics. So if you want to have a good understanding of philosophy of genetics, that's the place to go. He has published recently several papers in the Hastings Center Report, as well as the Lancet. And I will mention his two books. The most recent one is The Tyranny of the Gene, Personalized Medicine and Threat to Public, which has been widely reviewed, the Times Literary Supplement, Science, and so on and so forth. It's published in 2023 with Alfred A. Knopf. And in 2014, he wrote Beyond Versus, the struggle to understand the interaction of nature and nurture, with MIT, also widely reviewed in all the best places. His work has been reviewed and discussed in the New York Times and other important journals and magazines. And last but not least, is a former HPS graduate. So we are very proud. So Jim, the floor is yours.
Thank you, Eduardo. Thank you. I appreciate you giving me the chance to come out and talk with you all. This is a project that I'm working on with a graduate student who's wrapping up her PhD at Utah named Hannah Allen. So if you hear me say we, I'm referring to Hannah and I. And it's in paper form. We're basically just about ready to start sending it out to get some feedback and off to a journal. So this is a great kind of quality control moment to try out these ideas and see how they go over and incorporate any thoughts that you may have. So The talk I'm going to give today is basically about a story that's been told many, many times. And what I'm going to try to convince you of is, in the way that this story has been told all these times, it has repeatedly left this one rather glaring thing out. And so one of the goals today is to sort of tell that story again by putting that piece back in. But I think there's actually additional value to putting that piece back in. What I'm going to try to convince you of is, when we add that piece that has been left out, suddenly we can see some lessons from this well-trod path that have not been paid attention to in the past. And these are lessons that I think are particularly valuable for contemporary science, medicine, biomedical research. So that's kind of what I'm going to try to convince you of. So the story that's been told many, many times before is the Stateville Penitentiary malaria studies. This is a picture of the famous prison. And the Stateville Penitentiary malaria studies have been descended on by sort of two types of literatures. Okay. One type is largely research ethicists, clinicians, historians, reflecting on what in hindsight people widely agree was a really shocking series of experiments. So for about 30 years, research was done on prisoners in this prison outside Chicago. They were infected with malaria, and all sorts of antimalarials were tested on them to see which were the most effective. And as a result, this all happened from about the 40s to the 70s. There's this vast literature out there now that reflects on this, largely with an eye towards contemporary debates about whether or not consent can be ever really obtained in coercive environments. So, you know, a prison is obviously this carceral space, clearly coercive in nature. So can we really think of as informed consent, even if you get a signature or somebody saying, yes, I want to do that, is it true informed consent? And so there's a lot of literature on this that's using it as a case study to think about this balancing between consent and coercion. It turns out, though, that a number of the men, who we'll introduce you to shortly, who were given the antimalarials, had an adverse reaction to that drug. And the researchers who were doing the studies, in turn, saw it as an opportunity to try to get to the molecular bottom of adverse drug reactions. So why is it that for a handful of these people, when we give them the drug that we're giving everybody else, they have this really violent and what they initially thought was a deadly reaction to the drug? And it turns out that they successfully figure that out. And that becomes essentially the empirical foundation for what is later called pharmacogenetics and what we now call precision medicine. And so when people think about sort of giving, the punchline you often hear is giving the right treatment to the right patient at the right time, that you can trace right back from precision medicine to what was previously called personalized medicine to what was previously called pharmacogenomics and pharmacogenetics, and that all starts at Stateville as well. And so when people talk about sort of the history of this field, it often starts at Stateville. So these are the sort of two places where you'll find people talking about the Stateville Penitentiary malaria study. The standard story that you get here is that this research was all done on white prisoners. Okay. And what I want to, this is sort of the adding the piece back in that's often been left out. What I want to convince you of and draw your attention to is the fact that, in fact, over that 30 years, Black prisoners were extensively involved in the research. What's more, as it relates to this side of the story, when we look at what they actually experienced, there were all sorts of ethical problems and infractions that can't be easily boiled down to questions of consent and coercion. And it was actually their bodies, their blood, that was used to give us the data about adverse drug reactions, which then gets pharmacogenetics up and running. So the kind of the piece of the story that's going to get added back in is the story about the Black prisoners. And again, what I want to sort of ultimately then get to, this is where we'll end at the end of the talk, is to sort of focus on how, when we do add piece of the story back, we get these lessons, which I think are really important for some things that are happening in biomedical research and clinical practice today. So that's the kind of the big picture goal here. So the way I'll move through the material with you is I'll start by telling you this standard view of Stateville. If you were to go out and do an internet search for Stateville Penitentiary malaria study, look at the articles that are out there talking about it, this is what you're typically going to find. We'll then drop into what the researchers called primaquine sensitivity. Primaquine was the antimalarial drug that proved so effective, but also had this really violent reaction among the Black prisoners. And so the primaquine sensitivity is the thing that they're trying to figure out, which in turn sort of sets up what we're now calling precision medicine. And so I'll take a deeper look at that. And then finally, we'll take the step back and sort of take stock of, okay, now that we've added this part of the story back to it, what do we see that we didn't see before? So let's start with the standard view. The story of Stateville starts in World War II. So in World War II, access to natural supplies of antimalarials were disrupted by the war. And there were efforts all across the globe to try to come up with ways to create synthetic antimalarials. The way that played out in the United States was a number of different labs coordinated with a number of different prisons. All of this was funded by and overseen by the US military. And so the vast majority of it was happening at this prison just outside Chicago, about 40 miles southwest of Chicago. And so what was happening is antimalarials that were being synthetically manufactured elsewhere were shipped to the prisons. And then the clinical researchers there would use the prisoners to try out these antimalarials. So again, sort of, we're talking about the correctional facility is still there if you wanted to go tour Stateville today. It was largely overseen by Alf Alving. That's the kind of name who you see as the primary individual, although people like Ernst Bueller and Paul Carson were quite prominent as well. It's in this little town, Gillette. And what we should say is, by all sort of scientific standards, the research was incredibly successful. It starts in the 1940s, and by 1951, they know they've got this drug, primaquine, which is extremely effective. And in fact, it's still used all across the world as an effective antimalarial. The research during the war years, so we're thinking sort of mid-40s, was lauded publicly. So it was featured in a Life magazine article where the sort of story is all about, these sort of former enemies of society are now giving back to society, and what a great story this is. A couple things in particular, I think, also sort of made it really attractive to the media. One was Stateville had this really unique panopticon design. It was actually imagined and conceived by Jeremy Bentham. So all you utilitarians in the room, you can sort of thank him for what's to follow. He was imagining a sort of different way to monitor, to keep track of people. And so in contrast to the sort of old school model of prisons where you'd have these hallways down which all the cells were located and guard towers at the end, what Bentham imagined was a sort of big circle where all the cells are on the circumference. In the middle is a guard tower, but the prisoners can't see inside the guard tower. And so essentially, they never know whether or not they're being watched. And with just, 1 or 2 guards, you can keep track of hundreds of prisoners in a way that you wouldn't have been able to keep track of before. And so in the mid-20th century, this was seen as like a really novel, new technological approach to the criminal justice, to housing prisoners. So that's one thing that's sort of quite striking about it. The other is that Stateville had a very, very famous resident there, Nathan Leopold. So Leopold was one half of the Leopold and Loeb duo, who in the beginning of the 20th century were found guilty of committing what in the press was called the crime of the century. Essentially, they identified a young 14-year-old boy and decided to kill him just to see if they could get away with murder. They were convinced that they could get away with it. And they ended up sort of writing letters to the newspaper, and they thought it was going to be a big sort of game, and they were going to be so clever and sort of show the world how, you know, the geniuses that they were going to get away with this. One of them left their glasses at the site of the murder. They both end up in Stateville. Loeb was murdered in prison not too long after he got there, but Leopold, that's him there with at the time of the trial, and then this is him in the Life magazine article, spent the next 50 years at Stateville. And he was very involved in the Stateville studies. And so even after sort of the crime of the century is resolved, there's this ongoing interest in what happens at Stateville. And Leopold is sort of the lens through which the wider public sees that. And so, you know, again, sort of think mid-1940s, when people think about Stateville Penitentiary malaria study, it's wrapped in this aura of nationalism and wartime pride and giving back. And look, you've got somebody who committed this awful murder, but now look, he's actually sort of, you know, helping out with the studies. It's a feel-good story. That does not last very long. After the war, when the Nuremberg tribunals take place, to the surprise of the prosecutors from the United States, defense attorneys for Nazis introduced Stateville as part of the defense of what was happening in Nazi Germany. The argument was, right, hey, you're trying to hold all these guys accountable for what they were doing with people in concentration camps in Auschwitz and Dachau, all sorts of scientific experiments. But look, your own scientists were doing the same exact thing in Stateville at the same time. That naturally, rattled the prosecution, and they didn't have an easy answer. So what they end up doing, so this is Andrew Ivy. This is him testifying before Nuremberg and then him getting sworn in. He was a very prominent clinician and sort of face of medicine nationally, but also he was based out of Illinois. And so what was happening in Stateville sort of hit close to home for him. And so he was brought over to the Nuremberg tribunals, the trials, to make the case for why what was happening at Stateville was clearly different from what was happening with the Nazi scientists. And the argument that Ivy gives is, look, this is all about consent. Auschwitz, Dachau, these individuals had no say in whether or not they were going to participate in this research. Stateville, the prisoners were told what was going to happen, and they volunteered to do it. And so while on the face of it, it might look the same, they're both prisoner populations, this group opted in a way that this group didn't, and that's a fundamental difference. If you've ever read the Nuremberg Report, which is often sort of thought of as like one of the founding documents of modern research ethics, there's a really heavy emphasis on informed consent, and a lot of that is drawn from Ivy's testimony. And in turn, research in prisons continues in the United States for decades, bolstered by this idea that sort of Ivy has insulated us from the criticism that was being applied to the Nazis, right? As long as we got informed consent, it's okay. As long as they're signing off on this, we don't have to worry about this. That lasts for a little while, but you know, think, sort of imagine time machine, 1940s, fast forward to sort of mid-1970s. A couple things have changed. The war discussion is now no longer about World War II, it's about Vietnam, right? And so the national mood about military exercises and military-funded endeavors is quite different. The other thing that you're going to get by mid-1970s is Tuskegee syphilis study. Revelations of all sorts of sterilizations of marginalized groups across the United States. Bioethics as a discipline starts to come into its own. And so by the mid-1970s, the national mood about military and research involving marginalized populations has shifted quite dramatically. And so a number of these places where prison research was being conducted all sort of, you know, under the idea of, well, we've got informed consent, so there's nothing really to worry about here. Suddenly journalists descend on these places again, and we get a very different picture of what's happening, right? It's, wait a second here, you may have a form that has somebody's name on it, but these are clearly coercive carceral environments, and to say that somebody is sort of genuinely opting into this is just not a legitimate way of capturing the dynamic of what's going on here. And so in 1974, at Stateville, as well as a number of other places, prison research gets shut down. So these are just a couple of images from 1974 when the research was being shut down. That's a prisoner participant at Stateville. Then 50 years now since that's happened, you've got this steady sort of drip of reflection among scholars looking back on Stateville to use it as this case to think about this question of consent versus coercion. And what you can see is there's sort of two broad categories of perspectives out there. I'll mention the third in just a moment. On the one hand, there are quite a few defenders of what happened there. The idea is, look, they did volunteer, whatever you want to say about informed consent, like it was their decision and they volunteered to do it. You do have in newspapers, Leopold wrote an autobiography. You have these first person, what you might at first glance think are first person accounts of people who were participating in the study. And if you trust their words, they're saying, I was really happy to do this. I was honored to do this. I felt like I was giving back. This was something meaningful to me. Another thing that will be said is, we got to be careful about judging things that happened in the past based on concepts and sort of frameworks that we have today. So that we don't have the idea of a vulnerable research participant. You can't really sort of take them to task for not being extra careful with a vulnerable research participant. And then the last thing that you'll hear people say is like, look, the research was just really successful. Like, we got Primaquin. In a minute here, I'll talk to you about the Primaquin sensitivity. So that was really successful. And that should be part of this calculus as well. The critics, basically are willing to sort of grant those elements, but then say, but wait a second, like this was just a coercive environment. To participate in the research, you got access to financial resources that you wouldn't get access to anywhere else in the prison. You got access to healthcare opportunities that you wouldn't get anywhere else in the prison. The opportunity of getting paroled early was dangled in front of the prisoners by participating in the research. And so, yes, you might have a form with their name on it, but clearly the coercive environment in which these gentlemen were making this decision is not the kind of thing that you would sort of judge as being true informed consent. And then lastly, I'll just sort of point, a number of people have kind of said, it's complicated. It looks like we can all agree it was a coercive environment, but it also looks like some of the individuals were willing to say, like, despite all that, they were happy to do it. It's not the kind of thing that you can sort of clearly say one way or the other. What is not so much up for debate is who was involved in the research. The standard story is this was research that was all done on the white prisoners at Stateville. And I'm just giving you a couple of the quotes here that you sort of see. Right? The line that comes over and over again is sort of white, good physical health, had to be there for 18 months or longer. And you'll notice that a lot of these quotes, these are from some of the names that I had on that previous slide, are quotes of quotes. And they're quotes of quotes because what they're doing is pulling text out of the publications from the Stateville researchers themselves, right? And so I don't want to suggest that these folks were making this up. They were pointing to something from the primary literature and saying, hey, look, they were only doing it on white guys. So we can sort of put race aside. What we're going to focus on is this just fundamental question of when you're dealing with prisoners, can they consent? I want to do a sort of quick shout out to a couple people have said, wait a second, there were some black guys involved as well. That's Nathaniel Comfort. He had a paper in what we now call Studies in History and Philosophy of Science back in 2009. That's Harriet Washington, her book Medical Apartheid, which has been getting read a lot lately. Maybe you've come across it, has a chapter on prison research. Both of them, as part of larger narratives that they're involved in, largely talking about prison research, make it a point to say that at Stateville there were black participants as well. What they don't do, though, is sort of investigate in any way what actually happened to the black prisoners. And much of what I'm going to now start doing going forward is drawing your attention to how just saying like, oh, there were black prisoners involved in well, even that doesn't actually sort of do justice to what happened. We don't get the additional lessons just by saying, oh, there were black prisoners involved in well. It's what actually happened to them involved in the research. Okay, so now you've got hopefully sort of the sense of if you went out there and tried to sort of see what you can find out about the Stateville Penitentiary malaria studies, what you're going to get there. Again, it's basically research done on white prisoners, sets up the now sort of the contemporary debate that we can have about consent and coercion. As I mentioned at the sort of beginning of that section, by 1951, so just a few years after they get this up and running, they know that primaquine is a really effective antimalarial. They also know that in about 10% of Black prisoners, these individuals have a really violent hemolytic reaction to getting that drug. So by a hemolytic reaction, basically their red blood cells are breaking down faster than their body can recreate them. That leads to exhaustion, nausea, headaches. It does a number on your spleen, which is sort of in charge of trying to filter that stuff out. It looks to the researchers at this time as if they're having like just a really, really bad bout of malaria, actually. But it's not the malaria that it's doing it, it's the drug, the antimalarial drug that they're giving them. And so because primaquine is the drug that seems to have so much promise, the thought is, well, let's see if we can figure out why this particular subgroup is having this really violent reaction. What's going on? What makes them different at the biological level? And so starting around 1951, the research sort of bifurcates into two arms. They continue to do the ongoing antimalarial research for decades. And this is, again, largely done on the white prisoners. Where they're trying to see, they try out new synthetic antimalarials that come on. They try doing old antimalarials, but in new dosing regimens, right? Can you sort of give them more over a shorter period of time? Can you give it to them twice a day instead of once a day, right? What are the sort of basically most effective and efficient ways you can get this drug into people's body to prevent the malaria from taking over? But at the same time, then, they get this primaquine sensitivity research arm up and running, where they're going to be focusing on the Black prisoners who they know are primaquine sensitive and try to figure out what's going on there. And so these are the kind of questions that they're bringing to that primaquine sensitivity arm of the research, right? What is the inheritance pattern? How is this thing passed down from generation to generation? What's the underlying molecular mechanism that's giving rise to it? What happens if you just keep blasting somebody who's primaquine sensitive with primaquine? And ultimately, can they find a test? This was sort of like the grand prize. Can you find a test to determine in advance who is primaquine sensitive and who is not? And this really links up to what you can think of as like the big public health goal. And as they keep going back to the Army and asking for ongoing funding, this is the justification for doing that. Because primaquine seems so promising, there's a real thought of this isn't just going to be something that we're giving to troops in Korea now at this point. We could be doing worldwide anti-malarial campaigns. But if we're going to be rolling out primaquine all across the world, they figured based on their estimates that there were hundreds of millions of people who were likely to be primaquine sensitive. And so the goal was to have some sort of test that could go along with those anti-malarial campaigns so you could figure out who in advance is primaquine sensitive and then treat them differently, right? Give them the right treatment at the right time rather than giving what everybody else is being given and you risk poisoning them. And again, like the anti-malarial treatment that gives us primaquine, this was by all accounts, scientifically, very successful research. They end up figuring out that this is a sex-linked trait and it's passed down on the X chromosome. So much more common in men than women because men only have one X chromosome, with women getting two. Typically, you've got some level of buffer, even if you've got it on one of your X chromosomes. What's the underlying molecular mechanism? There is this G6PD gene, they now call it. But essentially what it does is creates an enzyme so that when your body is exposed to all the oxidative stress of being given the antimalarial, that is essential for cleaning that out and keeping your circulatory system functioning properly. When you don't have that G6PD gene and you don't have that enzyme, then the oxidative stress is what builds up, starts breaking apart your red blood cells, and that's where everything breaks down. It turns out they learned it was what they call now a self-limiting process. So when you give somebody who's primaquine sensitive primaquine, they go into that really violent hemolytic reaction. It devastates their red blood cells. But as their body creates new red blood cells, they are essentially immune to this process and they can slowly recover from it. And then finally, can a diagnostic test be created? Yes, in fact, several were created. And so I'm just giving you sort of like a couple of examples of the publications that came out here. And then notice, you'll see the names like there's Alvin and Carson, who I mentioned before. There's Alvin and Carson, who I mentioned before. There's Alvin. I don't know if Carson's on that one. This one starts with Carson, ends with Alvin. I put this one in a box because we're going to come back to it in just a moment. This was the one that came out in Science in 1956. That's the paper that actually sort of says like, hey, we found the gene that is responsible for primaquine sensitivity. We now call this G6PD deficiency. So, again, by scientific standards, this was really successful research. This is the research then, basically, that sets the foundation for what becomes pharmacogenetics and then later personalized medicine and precision medicine. That's Arno Matulski there on the left. He was a medical geneticist at the University of Washington at the time. That's Werner Kalo. He was a pharmacologist at the University of Toronto at the time. Both of them in the 50s going into the 60s were making the case to the scientific community about the value of trying to combine genetics and pharmacology. And what they were particularly sort of had their eye on were adverse drug reactions. The thought was these adverse drug reactions, many of them could be genetic in origin. And so for the geneticists, these adverse drug reactions could be really helpful case studies for tracing down the molecular mechanisms that give rise to it. And if you're on the pharmacology side of things, you don't want adverse drug reactions. So knowing more about the genetics is going to equip you with the scientific resources to try to make those things less likely. That gets called pharmacogenetics by the early 1960s. And what's important is there were very few actual case studies of somebody having traced an adverse drug reaction down to the genetic level at this time. The real explosion of these cases comes in the 80s going into the 90s. And so for the 50s, 60s, and then even into the 70s, there's not many case studies that people like Matulski and Kalo can point to say like, hey, we should be paying more attention to this. The one case that they do keep going back to is Stateville. Because Stateville was the one, it gets the Science publication, it's clearly G6PD deficiency. It's the sort of big success story that everybody can point to and say, we're going to be able to do this with other adverse drug reactions as well. This is a story that I tell in the book that Edouard kindly mentioned. Pharmacogenetics essentially is what gets repackaged as personalized medicine in the aughts. And then starting around 2010, gets repackaged again as precision medicine. What they all have in common is they're sort of in the business of delivering what they, again, sort of call the right treatment for the right patient at the right time. That's the lingo, that's the motto. I'm happy to talk about that in the Q&A if you want, but all I want to sort of get out here is the idea of like, you can sort of clearly draw this thread from Stateville to what everybody around the Pitt campus is excitingly calling precision medicine. Another way to just sort of think of this though is, here's a couple of publications that have come out in the last few years of clinicians talking about the history of their discipline, that being pharmacogenetics or personalized medicine or precision medicine. And what you see over and over again is them going back to that 1956 paper and saying, here is where it all starts, right? It starts at Stateville when they're getting to the bottom of primaquine sensitivity. Okay, so now to sort of pull it all together, I want to show you sort of what happened to the Black prisoners in order to generate that data that gave us those insights. And that's where now we're going to go beyond even what the scholars that have said, hey, there were some Black people involved as well, learn. And that's where the lessons really start to fall out. So, again, another way to think of this is like, what had to be done to people to get those answers? I'm going to mention 7 things that were done. One thing that's really striking is, think of this as a participant burden question. On the anti-malarial side of the arm, think of those 2 arms again, on the anti-malarial side, literally any prisoner in Stateville was a candidate for that. And so what you see is over the course of those 30 years, when they start in the 40s, ends in the 1970s, hundreds, more than 1,000 prisoners are involved in the antimalarial research. To be involved in the primaquine sensitivity research, you have to be primaquine sensitive. And so over those 30 years, by our calculation, we're only talking about tops 100 or so people. That means that they are involved in this research over and over and over and over again. And over and over and over again can mean being blasted with primaquine, even though you're primaquine sensitive, over and over and over again. Right? So this is just a sort of method section that we sort of pulled out of the first paper that kind of announces that they're investigating this. And essentially what they're telling you in the very sort of anesthetized, anesthetic clinical version of a method section is, we've got these guys and we gave them a really heavy dose of primaquine. And then after a few days, we were worried that they were going to die, so we took them off. When they recovered from that, we gave them a lower dose of primaquine. They had a reaction to it, but it wasn't as bad as the first. When they recovered from that, we gave them a really heavy dose of another antimalarial. And after a couple of days, it was so bad, we were afraid we were going to kill them, and we took them off that again. And like that's just one publication, one experiment, right? That these, particularly in this case, it was 5 prisoners were being exposed to. You don't see that sort of like, punch after punch after punch happening on the antimalarial side of it. The other thing that you see on the primaquine sensitivity side that you don't see in the antimalarial research is lots of identifiable information is included about the prisoners. So if you're participating in the research on the antimalarial efficacy side, you're going to find yourself in a publication as a case number. You're 1, you're 7, you're 12, you're 44. On the primaquine sensitive side, they often included initials, right? So this is WH, or sometimes the first several letters of a last name. And that's all in addition to things like age, height, gender, weight, right? Looking at any of these cases, when you've got initials and all that additional information, it's not hard to then make inferences about who in the prison is participating in this research and who isn't. And again, that's just not something that you see on the other side of this research. Another thing that ends up happening, because again, remember they're interested in like the inheritance pattern, is they start with the prisoners, but when they find somebody who's primaquine sensitive in an interesting way, like this gentleman HS, so he by all accounts tested as being primaquine sensitive, but was not phenotypically showing that response. They end up going out and recruiting his family members who aren't in the prison, right? Parents, siblings, trying to put together a pedigree to see how this is passed down. And as you can imagine, right, I mean, now we're talking about this coercive relationship extending not just into the walls of the prison. This isn't just something that a prisoner can sign off on. But you can imagine if you've got a sibling, right, in a prison in Pittsburgh, and somebody from the prison comes to you and says, hey, they're participating in research, would you be willing to participate as well? You're going to feel just as much pressure, perhaps more, than your loved one, because it looks like all those things that they're benefiting from are contingent on now you participating as well. The other thing you see is the research wasn't just confined to the prison. They were also trying to sort of see if they could develop, as I said earlier, tests for this. And so they go out into the public and recruit fairly large Black populations and large Black families and get blood from, in some cases, kids as young as 2 to see if they can do this, replicate this research on people out in the world. Another thing they do is go to the basically nearby prison reformatory for women, not too far from Stateville, to see if they can find out what's happening when women who are primaquine sensitive have this response. So remember, it's sex linked. Men, if they have it on their X chromosome, they're going to be affected. Even if a woman has it on one X chromosome, she's got the other X chromosome that can be protective. What they were curious to find and did find was a case of a woman who was affected on both X chromosomes. And what they found was her reaction was sort of the worst of anybody that they'd seen before. It was even worse than the men who were primaquine sensitive. But you only get that, obviously, if you're blasting somebody who's primaquine sensitive with primaquine. Another bit is we know from archive documents that Hannah found that the prisoners in the permaquin sensitivity side were paid about half as much as the gentlemen who were being paid on the antimalarial side. So despite everything I've said, also they're getting paid less for that work. And then the last one I'll mention before we sort of take a step back and talk about the lessons are Butler. He was one of the names that I mentioned at the beginning, Ernst Butler. He was somebody who continued to be, he became sort of like a president of the Hematological Society, a giant of hematology, a big name, and this all sort of comes out of his work at Stateville. And so he becomes really, really invested in permaquin sensitivity for years, even after he's sort of not involved in the army research anymore. And what he admits to the historian of science, Keith Whaley, who's at Princeton now, decades ago, is even after the prisoners were released, because that blood from the permaquin sensitivity men was so valuable, he would lure them back to his lab and give them some money in exchange for getting some blood from them. I won't read you this quote. This is sort of the point where he's describing it there. But what you'll see if you do decide to read it is, it's just a really crass description of his understanding of the relationship between him and the participants. There's this sort of sense of like, oh, you know, I was doing this research on these ne'er-do-wells in the prison, and I wanted to keep doing the research even though they were out of the prison. And look, I was like helping this guy out by giving him money, and then eventually he sort of runs off on my money and I never see him again. And like, of course, that's what you would expect when you're dealing with ne'er-do-wells who wind up in prison. So, you know, again, what I hope to have convinced you of so far is clearly Black prisoners were involved in the research. And what's more, when you were involved on the permaquin sensitivity side, we were seeing things that we weren't seeing when it was on the antimalarial side. So now what I want to do in the kind of last 5 to 10 minutes is draw out what I think are these sort of lessons that we learn now when we reflect on Stateville, focused on what was happening in the permaquin sensitivity side that hasn't been attended to when the story was told all the times before, and it was just a story about, you know, your average white prisoner being exposed to research in a coercive environment. The first thing is just to say, you know, again, the story over and over again at Nuremberg, when the prison research was shut down in the 70s, in the literature of the 50 years since, like the defining question is, can prisoners consent in a coercive environment? That's the ethical issue. Right? And then the back and forth is all about, well, how coercive was it? What was in the consent document? Right? It sort of boils down to this like consent coercion balance. And you know what I hope to have convinced you of is that when we sort of set that aside and just focus what was happening on the permaquin sensitivity side of things, there were all sorts of things going on over there that had nothing to do with just whether or not these guys could consent, right? Paying the permaquin sensitivity guys half as much as the antimalarial guys is not about consent. It's about sort of a two-tiered system where you're actually, you know, treating one group and rewarding one group for participating differently than another. Recruiting family members into this environment is not just about consent. It's about the sort of the social nature of vulnerability and how it can't be distilled down to like, you're this, you know, monad operating in free space and it's just a matter of whether or not you've signed the form or not. Like this sort of, it reaches out beyond you as an individual. Sharing identifiable information, it's not just about consent. This is about sort of seeing these participants as you can share information about them that you're not sharing about the folks over here. And so I think, you know, one just lesson is when we take this bigger picture of what happened, the fuller picture of what happened, I do think it's just harder to defend this, and even harder to say it's complicated, than when all you're looking at is the antimalarial side of things. Right? On that side of things, it's just, well, you know, we had this sort of research, it was coercive, but also they consented, you know, you can sort of do that calculus yourself, maybe we disagree. The stuff that was happening over the permaquin sensitivity side, it clearly had scientific purpose. Right, they weren't just like poking these guys for the hell of it because they thought it was, you know, they were sort of sadistic. But it's much more than just about consent, and I think it's harder to justify. The second thing that the lesson that I want to draw out, and this is one that I think very clearly is something that we should be mindful of today. go back and remember the original idea for doing the permaquin sensitivity research, the deliverable that everybody was excited about, the thing that sort of kept the military coming back and investing more money, was this idea that the Black prisoners who were permaquin sensitive were in some way representative of that target population of permaquin sensitive people all over the world. And so if we can get to the bottom of this with this research group, we can have that thing, namely the test, to deploy out so that people with permaquin sensitivity everywhere can be treated appropriately when we're trying to roll out an antimalarial campaign. So it turns out now that the World Health Organization recommends this. They say, if you're going to do an antimalarial campaign, you should do G6PD deficiency testing as part of that to make sure that you're treating the people who are permaquin sensitive differently than the people who are not. But this is drawing on a paper that just came out in the journal Pathogens last year. It turns out in the vast majority of regions where malaria is endemic, this just doesn't happen. It's not that it doesn't happen because the science is bad or that there aren't any tests. The science is good, there are tests, but it doesn't happen because of all the logistical, financial, social stuff that happens after a scientific discovery is made. When you try to translate that into actually going out and helping the people that you said you were going to be helping, everything is tilted against actually delivering that for people who are permaquin sensitive. So there are all sorts of financial barriers to having these tests available when they're doing antimalarial campaigns. There's just like logistical barriers when it comes to processing the tests and getting them back in time. There are training barriers so that the people who are out there doing the antimalarial campaigns can understand how to use the test, right? And the lesson here that I take from this is, you know, you may have that legitimate good intention to sort of say, hey, we're going to study you because we're going to be able to help all these people like you. But really being able to deliver on that is not about sort of getting that data from those individuals. It's about operating in a world that's tilted against trying to help people that don't have many resources. And there's this kind of cool irony here for precision medicine today. You in Pittsburgh, in Oakland, are on the front lines of the precision medicine industry, right? I just took a Google snapshot of this All of Us recruiting office, right? Just on Forbes Avenue. I don't, sort of like just a couple blocks that way, right next to the Starbucks. The All of Us research program was sort of rolled out primarily targeting communities of color, right? So they come here and they're really looking at the trees in the Hills neighborhood. They go to New York, they're based in Harlem. They go to Birmingham, they're reaching out to the Black communities there. The pitch that the All of Us research program made was all about, we're going to access diverse communities, get diverse data, and then combat the health disparities that are impacting those communities. And there's just like, in part, it's justified on the idea of precision medicine hasn't been good about delivering on the promises to communities of color. It's been tilted towards white people. And so the All of Us research program is going to switch that around and really repurpose it for communities of color. And the first thing to say is just this is like really weird irony in the fact that they're trying to now target communities of color so that they can benefit from precision medicine. And yet the whole thing started with this vulnerable population of Black male prisoners in Stateville, right? The more general lesson that I just want to draw is this point that just because you have diverse data from a diverse community, in no way guarantees that you are actually going to be able to deliver the health benefits to that diverse population or to that diverse target group that you say you are interested in. And much of all of us, and I would say much of medical genetic research more generally now, you see, I go to some of these medical genetics conferences, and there's just all this emphasis on getting diverse data, and people sort of, they applaud themselves when they've got diverse data, and they talk about how successful they've been in recruiting a diverse research cohort. And it's sort of like this idea of once we get the diverse data, we will, sort of like snap our fingers or wave a wand, and then all the research outcomes are going to go back and help that diverse community. And I just think there's plenty of reason to think that that's not the case. And I think Stateville is a great example, sort of where the whole story starts here. And yet 50 years later, we're still not doing the thing that they said that they were aiming to do, which was sort of have this test that you can have people who are chemical sensitive when they're getting the antimalarials. And then the last thing I'll mention, this is a real quick one, is just, I think it's worth saying, there's all sorts of places where you can sort of see the footprints of Stateville. And we've been talking about many of them, right? There is now ongoing talk about making research in prisons more accessible. It really got shut down in the 70s. There are concerns that now limiting research in prisons actually sort of cuts prisoners off from the benefits of research. And so there's a movement more in the direction now of trying to open that up again and letting them participate. When those conversations happen, this is partly about Stateville. I think you could think of this at the clinical level, when an oncologist sort of is biopsying a tumor and telling a patient, hey, we're going to genotype your tumor before we decide on which course of treatment. That is what the oncologists would call targeted therapies or precision medicine. That is pharmacogenetics, that is Stateville. When a pharmacogenomicist, tells a class of medical students, let me tell you all about right treatment for the right patient at the right time. I'm sure you all have heard that. That's the precision medicine motto. This goes back to Stateville. And then again, when you've got these authors who are sort of pointing to the 1956 Carson article and saying, hey, this is where precision medicine begins, that's about Stateville. But the real lesson that I see it is, we've sort of forgot these Black prisoners who were involved in it. They were the ones who were experiencing all these awful things. They were the one giving their time, their bodies, their blood, that gets the data, that gets all this up and running. And so I think there's a real case to be made for sort of having a way to acknowledge them when we're talking about these various applications. So again, just to wrap up, standard view, hey, this awful thing happened. We should be talking about consent and coercive environments. Hey, look, we get right treatment for right patient at the right time. That all goes back to Stateville. This involved white prisoners. Trying to draw your attention to the fact that there was this other arm of it that involved the Black prisoners, and I think there's lessons that we could sort of glean from when we pay attention to those. And again, this is a project with Hannah Allen. It's been funded by the NIH, and I thank you all for listening.
Take a 2-minute break, and then we'll have time for questions.
First in line. Do I need to do it?
I should get